B.Sc second, semester 3, paper 6, CBCS syllabus is plant physiology. In this unit number 2 is photosynthesis. Today we will see about photosynthesis and photosynthetic pigments. We will start to learn this topic, photosynthesis. First we will see about introduction and significance. In the unicellular or multicellular life form, proper organization is necessary to carry out all the life processes, utilizing available energy in the system. During such processes, complex organic molecules are synthesized for conservation of energy, and organic molecules are broken down for release of energy. All the living systems therefore need energy for their existence. The three basic requirements for the existence of life in different organisms are water, light and air. The source of visible light is the sun, and the sunlight energy is the only energy used during energy transforming reactions in all living systems. Photosynthesis is the only process which can fix the sunlight energy and conserve it in the form of carbon compounds. These carbon compounds are used by all other living forms as the source of energy required for the maintenance and replication of life. Therefore, photosynthesis in green plants is the most important physico-biochemical process. The existence of life is dependent on the photosynthesis. Photosynthesis is the ability of the green plants to fix and use the sunlight energy for the synthesis of carbon containing organic substances from inorganic raw materials available in the environment. The organization of photosynthesis in green plants is in the form of photosynthetic apparatus. The photosynthetic apparatus consists of different photosynthetic pigments and other chemical substances. In all these pigments, chlorophyll A is the only pigment which can take direct part in photochemical act, that is, fixation and conservation of light energy. All other pigments act as accessory pigments and help the chlorophyll A molecule to carry out and complete the photochemical act. The photosynthetic pigments in photosynthetic apparatus are present in form of pigment systems or photosystems. In such pigment systems, reaction center complexes are present and they carry out photochemical reactions. The discovery of immersion effect confirmed the presence of two pigments systems, PS first and PS second, in photosynthetic apparatus, which carry out photophosphorylation and conserve the fixed light energy in ATP molecules. The synthesis of ATP molecules takes place by electron transport mechanism. The mechanism is also responsible for the synthesis of hydrogen donor molecule in form of NADPH and ATP molecules. Both ATP and NADPH molecules are essential for assimilation of CO2 during carbohydrate synthesis. These two molecules together form assimilatory power for carbohydrate synthesis and are synthesized by fixation of light energy. For the synthesis of carbohydrates utilizing assimilatory power molecules and atmospheric CO2, in the different plants, three different pathways are followed. C3 plants follow Calvin cycle pathway. C4 plants follow HSK pathway, Hatch, Slack and Kortschak pathway, and some succulents follow CAM or crassulacean acid metabolism pathway. In this slide, we will learn about photosynthetic apparatus. To carry out all the reactions in photosynthesis, from fixing of sunlight energy up to synthesis of different types of carbon compounds, proper organization is essential. Such organization is found in a photosynthesizing cell in form of specific structure embedded in cytoplasm. In simple, prokaryotic photosynthesizing cell, photosynthetic bacteria, blue-green algae, such structures are present in form of free or isolated photosynthetic lamellae called chromatophores. These chromatophores represent photosynthetic apparatus of simple primitive nature. But in case of bryophytes or vascular plants or higher plants, well-developed evolved plants, complicated photosynthetic apparatus is present. Such photosynthetic apparatus is in form of green plastids embedded in cytoplasm known as chloroplastids. So in all higher plants, bryophytes and eukaryotic algae, photosynthetic apparatus is chloroplastid. Let us see this figure. It is structural regions of chloroplastid, which shows double layered chloroplast envelope, matrix, internal system of membranes, thylakoids, interthylakoid space or lumen, stroma thylakoid and grana regions. The chloroplast envelope shows two membranes, each about 5 to 7.5 nm thick, and are separated by about 10 nm intramembrane space. Out of these two membranes, the inner is selectively permeable and it regulates the exchange of metabolites between chloroplastid and cytoplasm. The envelope encloses stroma of chloroplastid. The stroma in chloroplastid is a protein solution with many substances required for photosynthetic reactions. This region contains all enzymes required for photosynthetic carbon reduction, that is synthesis of carbohydrates from atmospheric CO2. The stroma region is with complex lamellar system of membranes. This system is composed of individual pairs of parallel membranes which appear to be joined at the ends. The sectional view of the chloroplastid shows double layered structures, flattened sac known as thylakoids. Thylakoids are arranged one above the other, forming stacks or tiers known as grana. The thylakoids in granum region are called as grana thylakoids. Some thylakoids extend beyond grana region into stroma region and known as stroma thylakoids. Such thylakoids connect two grana regions and form interconnection between different grana regions. The chloroplastids show presence of grana regions. Some are without grana. For example, chloroplastids in parenchyma bundle sheath cells surrounding vascular bundles in C4 plants. This shows that chloroplastids are of two types, with granar region granar and without granar region agranar. The interior space of thylakoid is called lumen. It is the site for photolysis of water or evolution of photosynthetic O2 takes place in lumen. The main function of lumen is to store protons which are pumped across the thylakoid membrane during electron transport. These protons are used to drive ATP synthesis during photosynthetic light reaction. In granar chloroplastids, photosynthetic pigments are present in granar region. In agranar chloroplastids, they are present in tube-like thylakoid structures. The section of chloroplastid passing through granum region under electron microscope shows the granum thylakoid envelope is double layered like chloroplast envelope. The double layered envelope shows some particles on lower surface of the outer membrane and on the upper surface of the inner membrane of thylakoid. The particles on the surface of outer membrane are loosely arranged and on the inner membrane are compactly arranged. These compactly arranged particles on outer surface of the inner membrane of thylakoid envelope are called as contasomes. These contasomes are considered as sites of photochemical reactions. In these contasomes, photosynthetic pigments are present to carry out reactions essential for fixation of light energy. Then it conserves ATP molecules. Each contasome under electron microscope shows 4 subunits, contasomal subunits. Each contasomal subunit shows presence of many molecules of photosynthetically active pigments. Contasomes consist of pigment molecules and these molecules are arranged in definite system called pigment system or photosystems. As per nature of the pigment, photosystems are of two types, photosystem first and photosystem second. Let us see this figure which shows contasomes on the surface of thylakoid membrane. Thylakoids form granum. Thylakoid showing two membranes of thylakoid envelope. The contasomes are arranged on outer surface of inner membrane and contasomal subunit are with pigment systems. The inner thylakoid membrane shows presence of contasomes with photosynthetically active pigments on one side towards stroma and presence of lumen which is with protons on other side. During fixation of light energy, the stored protons in lumen are pumped across the surface of thylakoid membrane having contasomes. By using these protons, ATP are synthesized by electron transport process. The lumen of thylakoid is the site for photolysis of water and evolution of O2 during photochemical reaction. So lumen is on one side and pigments taking part in the fixation of light energy are on the other side. The synthesis of assimilatory power ATP and NADPH2 takes place on the surface of inner membrane of the thylakoid in the granar region of the chloroplastid. The granathylakoids complete the photochemical act and synthesize assimilatory power molecules. The stroma region completes all the reaction required for CO2 assimilation, utilizing assimilatory power during carbohydrate synthesis. Both the major steps, light reaction and CO2 assimilation, are completed inside the chloroplastid. So, it is the proper organization for photosynthesis in the cell and it is considered as a photosynthetic apparatus. Then we will study about photosynthetic pigments, chlorophyll, carotenoids and phycobilins. Photosynthetic pigments are produced during photosynthesis. Energy from sunlight rays is trapped and it is converted into chemical energy using these pigments. This process is called photochemical act or photochemical reaction. The pigments which take part in the photochemical act are considered as photosynthetic pigments. These photosynthetic pigments are present in the chloroplastid. These pigments are classified into three groups: chlorophylls, carotenoids and phycobilins. First, we will learn about chlorophylls. The photosynthetic apparatus in different types of the plants shows that there are many types of chlorophylls in plant cells. These different types of chlorophylls are chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll bacteriochlorophyll A, chlorophyll bacteriochlorophyll B, chlorobium chlorophyll. A molecule of chlorophyll is chemically porphyrin derivative. It shows two regions like head region and tail region. The head region shows tetrapyrrole ring with atom of magnesium in the form of nucleus in the center of the ring, and the tail region shows phytol chain. The chemical formula of chlorophyll A is C55H72O5N4Mg and chlorophyll B is C55H70O6N4Mg. In a chloroplastid, most of the chlorophyll molecules are present in the form of protein complex and few molecules are present in the free form. The chlorophylls in the protein complex are useful for photochemical act, while free chlorophyll molecules are harmful as they are responsible for photooxidation. It reduces the percentage of chloroplastids. All photosynthetic organisms which evolve O2 during the process of photosynthesis shows presence of chlorophyll A in their photosynthetic apparatus. In photosynthetic bacteria, bacteriochlorophyll A is present. Chlorophyll B and chlorophyll A is present in the photosynthetic apparatus of higher plants. And in the member of two divisions of algae, chlorophyta and euglenophyta, chlorophyll A and chlorophyll B is present. The chlorophyll C and chlorophyll A is present in marine plants. It is present in members of algal divisions, Phaeophyta, Pyrophyta, Bacillariophyta, Chrysophyta. Chlorophyll D is present in algal members of Rhodophyta, while chlorophyll E is present in division Xanthophyta. In the group chlorophylls, only chlorophyll A can take direct part in the photochemical act, while all other chlorophylls are support chlorophyll A to carry out photochemical reactions. So except chlorophyll A, all other chlorophylls are minor pigments and called as accessory pigments. These are supporting pigments. The chlorophyll A molecule, which takes direct part in photochemical act, exists in different forms. These forms absorb light energy from different zones in the visible spectrum region. Chlorophyll A P700, chlorophyll A P680 and chlorophyll A P660. These are the important forms of chlorophyll. In this slide, we will study about carotenoids. The carotenoids form second major group of the photosynthetic pigment, which are chemically terpenoids. It shows two types, hydrocarbon carotenoids and their oxygen derivatives xanthophylls. The hydrocarbon carotenoids are orange or red orange pigments and xanthophylls are yellow colored pigments. Beta carotene is the major carotene in algae and higher plants. In some plant, alpha carotene is also present. In bacteria, gamma carotene is present. The important xanthophylls are lutein, luteol, zeaxanthin, phylaxanthin, neoxanthin. Carotenoids occur in the form of protein complexes in the photosynthetic apparatus and their presence play important role. They protect chlorophylls from photooxidation. They absorb light energy and transfer that energy to chlorophyll during photochemical reactions. Carotenoids cannot take direct part in photochemical act and therefore they transfer the absorbed light energy to chlorophyll A molecule. Due to such supporting role, these are considered as accessory pigments. Here we will study about phycobilins. The name phycobilins consists of word phykos means algae. So the name phycobilins indicate the algal origin of the pigment. Chemically, these are open chain straight tetrapyrrole. As these pigments form integral part of protein molecules, they are also considered as a chromoproteins. The tetrapyrrole colored portion is called as a chromophore, which absorb light energy. These photosynthetic pigments occur in algae in the division Rhodophyta, Cyanophyta, Chrysophyta. The important phycobilins are red colored phycoerythrins and blue colored phycocyanin. These pigments along with chlorophyll A and carotenoids take part in photochemical reaction. Like carotenoids, phycobilins absorb light energy and transfer it to chlorophyll A, so considered as accessory pigments. See this chart, chlorophylls. Chlorophylls occurs in different plants, algae, diatoms. Chlorophyll A present in higher plant and algae. Chlorophyll B is present in higher plants and algae. Chlorophyll C is present in diatoms and brown algae. Chlorophyll D is present in red algae. Carotenoids. Alpha carotene is present in higher plants and algae. Beta carotene is present in plants and algae. Luteol present in red algae higher plants. Phyllosxanthol present in higher plants. Fucoxanthol present in diatoms or brown algae. Phycobilins are present in algae, blue-green algae, red algae. Phycoerythrins are present in red algae, blue-green algae. All phycocyanins are present in blue-green and red algae. Then we will see about accessory pigments. During photochemical reactions, an excited electron with extra amount of energy is ejected from chlorophyll A molecule. From this ejected electron, extra amount of energy is released to bring back the electron to its normal energy level. This energy is trapped and conserved in ATP molecules with the help of electron transport system in photosynthetic apparatus. The chlorophyll A molecule take direct part in photochemical act. For the synthesis of ATP molecule by trapping energy, an electron constant supply of specific amount of energy to specific chlorophyll molecule A is essential. To maintain continuous constant supply of specific amount of energy to chlorophyll A molecule requires photosynthetic pigments. So all photosynthetic pigments function together to supply more energy to chlorophyll A, since only chlorophyll A can take part in photochemical act and all other pigment molecule cannot take direct part in the energy absorbed by all these pigment molecules is transferred to chlorophyll A molecule. All such pigment molecules which help the chlorophyll A molecule to complete photochemical act are called accessory pigments. Chlorophyll B, C, D, E, carotenoid, phycobilins are accessory pigments. Due to these accessory pigments, light energy from the entire region of the visible spectrum can be absorbed and used for the photosynthesis. Then we will see about photoprotective carotenoids. The thylakoid membranes contains pigments, protein, chlorophyll complex molecule and carotenoid complex molecule, which are essential for proper transfer of energy during chemical act. The carotenoid containing molecules carry out two functions: harvesting of light energy and photoprotection of chloroplastid. Studies with albino white seedling and carotene deficient seedling indicated that absence of carotenoid in the cells in aerial organs lead to photooxidation of chlorophyll. In photosynthetic apparatus, the molecules of chlorophyll required absorption and transfer of light energy in form of pigment protein complex molecules. Some molecules of chlorophyll exist in free molecule form, which are dangerous. It is harmful because if the energy absorbed by these free molecules is not utilized properly, it carries photooxidation of pigment. Many plants, because of their genetic nature under high light intensity conditions, absorb relatively more light energy than their capacity to utilize for photosynthetic CO2 reduction process. If this excess amount of energy is not removed by proper mechanism, it leads in photoinhibition or photodamage or destruction of photosynthetic apparatus in the photosynthesis. Many plants show link between removal of excess energy and presence of xanthophyll zeaxanthin. By a process known as xanthophyll cycle, zeaxanthin is synthesized from xanthophyll phyloxanthin. In this xanthophyll cycle, phyloxanthin is converted into zeaxanthin. Two steps with the production of anthraxanthin as intermediate compound. The xanthophyll zeaxanthin produced in this cycle forms zeaxanthin protein complex molecules by binding itself to protein in photosystem second. The xanthophyll, anthraxanthin and zeaxanthin absorb excess amount of energy from chlorophyll molecule when it is not used in photosynthetic energy transfer reaction. These two xanthophylls have capacity to dissipate the excess light energy as heat energy. The xanthophyll cycle starts when light energy is in excess and it stops when there is no excess energy. The cycle acts as an effective switch to avoid bad effects of light energy. The importance of xanthophylls or carotenoids as photoprotective pigments has been observed in the plants in the form of changes in total carotenoid content as per the change in environmental light conditions. For example, plants growing in sunlight condition are with more carotenoid and less chlorophyll in the leaf cells, and plants growing in shade exhibit total amount of chlorophyll which is more than carotenoids in the leaf cells. So in shade plants, leaves consist of more chlorophyll, and in sunlight condition more carotenoids are present and less chlorophylls are present in the leaf cells. Thank you.